I will talk about functional effects of mutations, not just about epilepsy, but also about FHM, familial hemiplegic migraine. We talked a little bit earlier with Jeff Noble about it. So when we do studies of functional effects, we are lucky. We are lucky because we have, thanks to the clinicians and the geneticists, we have a phenotype and a clear cause of the disease, more or less. That is the pathogenic mutation. So when you get the functional effect, you can close the loop, basically. Everything should be clear. You have the phenotype, the cause of the disease, the functional effect. You could target the functional effect for therapeutic developments. But there are several layers of complexity here. But one of them is that we study the functional effect with experimental systems. They are limited, as I will show you. And then the pathogenic mutation often is just the initial cause. And the effect that you see studying this single pathogenic mutation is the initial functional effect. Then the biological system is perturbed and it responds with homeostatic responses and pro-pathologic responses, so pathologic remodeling. So you change different things in the system till you get to the mature phenotype. So to understand the mature phenotype, you need to understand, yes, the initial functional effect, the functional effect of the genetic mutation that you have identified on the target protein, but then also all these responses that can be different in comparison with the initial functional effect. So, as I said, we use experimental systems that have limits. One experimental system are transfected mammalian cultured cells. Here, you express your protein of interest, the mutant and the wild type. You compare the properties, and so you can identify the effect of the mutation. For the sodium channel, you can record sodium currents with electrophysiological recordings or action potentials, so the excitability if you are using neurons, because you can use cell lines that basically don't have other ion channels, and you study the properties of your ion channel in isolation, or you can use neurons, and so you have also all the other ion channels expressed there, and you can identify the effect on excitability. So these models model the initial cause of the disease, so the functional effect on the mutated protein. And they allow a relatively large screen of mutations for functional studies. But as I said, there are several pitfalls, so you have to use them with caution because they are very reduced systems. So you cannot compare it with an organism, you cannot compare it directly with a patient. Then you have in vivo models, for example, rodent models. Then you can engineer the mutation in the genome of a mouse, for example, and study the effect of that mutation. And here you can model the initial cause of that mutation on the target protein, but at different levels, molecular, cellular, network, behavioral levels. And these models, they model also homeostatic responses and pro-pathologic remodeling that lead to mature phenotype, as I told you before. But they don't allow relatively larger screens of mutations because they are difficult to develop, costly. They are anyway models, so they don't have the same properties, features of the patient. So you should use them anyway with caution, even if the caution here is smaller than for more reduced experimental models. So these are the initial mouse models of Dravet syndrome caused by SCN1A mutations, as we know here. They were constitutive, gene-targeted mouse models carrying truncated mutations. So the mutant protein was non-functional. The first one was the knockout NAV1.1 that we developed in Bill Carter's lab. The other one was developed by the group of Yamakawa in Japan. And they have, these two models, they have basically the same phenotype. They carry a similar mutation. And so with these two models, we identified the initial effect of these mutations, like that was hypoexcitability of GABAergic neurons caused by this loss of function, complete loss of function of NAV1.1. And we identified a reduced inhibition in neuronal network that was consistent with the phenotype, seizures. Then now there are several other models. There are also conditional models. These are called the Kree-Lox or Flox models. These are Flox knockout mouse models because you normally don't have the mutation expressed. You have the mutation expressed just if you expressed in these mice Kree recombinase. You can express Kree recombinase in different subtypes of neurons or in different developmental periods. So you can identify the effect of the genetic mutation in different subtypes of neurons, for example, like in these papers, or in this one in particular, in which the effect of parvalbumin interneurons, GABAergic interneurons, was highlighted. So these models are knockouts. So you block completely the function of the protein. There are also models of missense mutations. Missense mutation changes a single amino acid in the protein. You have still some of the function of the protein. This is a GEF plus model carrying this mutation, the R1648H. This has been developed by Marian Meisler, and I think inherited by Andrew Escari. This is a study that we performed with Holger Lerse. And we showed here that this partial loss of function can anyway induce reduced GABAergic excitability, mainly because of dysfunctional axon initial segments. And this can cause hyperexcitable networks in different brain areas, from the hippocampus to the cortex to the thalamus. And now this is the last one, I think, on the market. This is a Floxed knock-in of a Dravet syndrome mutation. So it's a missense mutation that causes in a patient Dravet syndrome mutation, and it has been developed by the Dravet Syndrome Association. It is freely available. Now, I will switch a little bit on migraine, familial hemiplegic migraine type 3. This is caused by a missense mutation of NAV1.1, SCN1A. It's a rare, severe form of migraine with aura that is characterized by hemiplegia during the attacks. These patients in particular don't have seizures linked to the migraine attacks. Epilepsy in general is very rare in this patient. So it's a kind of a pure hemiplegic migraine phenotype. So we studied several mutations. I will show you a couple here. Using these reduced systems that are human cell lines, transfected human cell lines, or transfected neurons. So in the beginning, when we expressed the mutation, we got this. So in black, you see the wild type, the current amplitude, basically, of the wild type. In white, the current amplitude of the mutant. So basically, no current, complete loss of function, like for a Dravet syndrome mutation. That was a kind of puzzling. So before that, this was a folding defective mutant. Folding defective mutants are mutants with that cannot acquire the correct three-dimensional structure. So they are recognized as unfolded defective proteins and degraded by the quality control system of the endoplasmic reticulum of the cell. But then, in certain conditions, you can rescue them. For example, when you incubate cells at 30 degrees, a lower temperature. And so when we did that, we rescued it. So we got current. We got basically 50% of the current in comparison with the wild type. This is the current. That is a bit weird, as you can see there, if you compare the mutant and the wild type. And we were able to rescue it, not just with temperature, but also with other proteins that interact with NAV1.1. For example, ankyrin G, you have heard about it this morning, calmodulin. And so this mutant that looks like a complete loss of function mutant is actually rescuable, and you have current. The interesting thing is that when you express it in neurons, you don't need to incubate them at 30 degrees. You don't need to have these rescuing approaches to have current, because you have current directly there. So something in the neurons can rescue this mutant. And when you have current, you have more current than for the wild type. You can see here the difference between wild type and mutant. You have more current here. And when you record the action potential discharges of these neurons, transfected neurons, you get more firing, higher firing frequency when the neurons are transfected with the mutant in comparison with the wild type. So the mutant is a gain of function mutant. And we have now another FHM type 3 mutant, Missense Mutation, that shows similar properties. So we have two mutants that look like completely loss of function, but actually they are gain of function mutants. And so when we compare with epileptogenic mutants of NAV1.1, and we did here a screen in the same conditions that we used for the migraine mutants, we never get gain of function. So we can partially rescue them, but we never get gain of function. We get gain of function just with the migraine mutants. So we think here, another thing. I say this first. We think here that loss of function of NAV1.1 causes epilepsy, a gain of function causes migraine. This is another pathogenic mechanism that you can study in this reduced system, negative dominance. So it's the interaction between the wild type and the mutant, and this interaction can decrease the function of the wild type, or the co-expressed wild type. So you have an effect on the wild type co-expressed in the same cell. And in the beginning, we published a paper in which we found no negative dominance for epileptogenic NAV1.1 mutants, but now we found some of them that have this pathogenic effect. You can take a look at the poster of Benedetta Terragni here. So I go back to loss of function, gain of function, epilepsy, migraine. But how can NAV1.1 gain of function can lead to migraine? So our hypothesis here is that gain of function of NAV1.1 can induce hyperexcitability of GABAergic neurons. And so this can, in our hypothesis, induce a pathomechanism of migraine, that is cortical spray depression, that you have heard about this morning. That is a hyperexcitability of cortical neurons followed by a long-lasting depolarization, depolarizing block, so silencing of fire. So we tested this hypothesis, that would be another mechanism of cortical spray depression induction, with optogenetics. So optogenetics, we can control with light the activity of neurons, or different types of neurons. So we controlled here the activity of GABAergic neurons, and we induced hyperexcitability with blue light of GABAergic neurons in a selective way, just GABAergic neurons. And when we do it, we can apply spatial illumination, so just in a small area of the cortex, and we can get cortical spray depression that we can monitor with electrophysiological recordings like field potential here. This is the electrographic marker, basically, of cortical spray depression. And with imaging, you see with imaging this wave in the spreads in the cortex, that is the wave of cortical spray depression. And now, if it works, you can see here. Here we are applying blue light, so we are exciting GABAergic neurons. And eventually, you get cortical spring depression, this wave here that you see in the video. So this was done in a brain slice. We get the same thing in vivo, in a live animal. You can see here we apply blue light, and we record electrophysiologically this CSD signature here. So this is a novel mechanism of cortical spring depression and of migraine. It's a different mechanism than in epilepsy. Now, this is another experimental model. These are human neurons that you can differentiate from induced pluripotent stem cells. Basically, you get just a skin biopsy from a patient, and then you can get the human neurons from that patient. This model, but it's really cool because then you have human neurons of your patients, so you can do a bunch of things from disease modeling to cell therapy to drug discovery. It has been used for studying the functional effect of Dravet syndrome mutations. And the first paper came out was this one from the Michigan group, in which the authors found increased sodium current and hyperexcitability of both glutamatergic and GABAergic neurons. So this is different than what you see in the cell lines, in the transfected neurons, or in the mice. And that was puzzling. Then a second one came out, similar approach, and here they found hypoexcitability of GABAergic neurons, like in the mice and the cell lines. So here they were 1-to-1. Then the history is following paper found hyperexcitability of glutamatergic neurons, again against the results obtained with other systems. But then another one found hypoexcitability of GABAergic neurons. Here we are 2-to-2. Then another one here, 2016, hypoexcitability of GABAergic neurons. This was a kind of elaborated study with a lot of controls. Anyway, here they found hypoexcitability just of inhibitory neurons. And this one, again, 2018, just hypoexcitability of GABAergic neurons. And here, we are here at 4-to-2. So which study is correct? So this is our experience. For sure, here you have a lot of variability. We tried as well to study Dravet syndrome mutation with the system, and we got a lot of variability. That depends on the cultural conditions, on the maturity of the neurons. Sometimes you get great neurons that fire very well, but if the neuron is not well mature, it doesn't fire very well. So in our hands, there was a lot of variability. But these differential effects depend just on variability, are only a problem of experimental conditions. Maybe not, because here maybe you model not just the initial cause that you see in these cell lines or initially in mice, but also the other responses of the system, propathologic, homeostatic. The hypothesis here that is that NaV1.1 loss of function can lead to different effects on neuronal excitability because of these different responses of the system. So we search for homeostatic responses. We compared these two neurons, GABAergic neurons. Number 2 is parvalbumin positive basket cells that have been studied for a long time now, in which you have always hypoexcitability. But we studied also this number 4 here. They are cholecystokinin positive neurons. And we did it in the classic NaV1.1 knockout Dravet mice. So we found, as everybody, decreased excitability of these parvalbumin positive interneurons. But in these CCK neurons that we selected because they are very much modulated, much more than the PV neurons that are very rigid, CCK neurons are very plastic. And we found in these neurons increased excitability. So the opposite effect in comparison with PV-positive neurons in the same animal model, in the same brain structure, so the hippocampus. So we found an homeostatic response in CCK basket neurons, but not in PV basket neurons. And this is a long story, I will make it short. We thought that an important player in this homeostatic response was cholecystokinin, that is co-released with GABA from the synaptic terminals of these neurons. A cholecystokinin can increase the excitability of other interneurons, for example, the parvalbumin positive ones. And we thought that this was an important homeostatic response. In fact, when we have hyperinsufficiency of cholecystokinin, because here we crossed the SCN1A heterozygote knockout mice with heterozygote knockout mice for cholecystokinin, so I have a double heterozygote. In the double heterozygote, you get higher mortality, more severe seizures, and longer seizures. So this is an important homeostatic response that that can modify the phenotype of the mice. And so if you want to know more about it, there is a poster here, and Paolo Scarmani will present it. So the last slide, basically. This is another story about another response. This response is a pathologic remodeling that we have found in GEF+ knock-in mice that I told you about in the beginning. So we used these mice that have a very mild GEF+ phenotype. Basically, they almost don't have seizures, to induce seizures because they are sensitive to hyperthermia, they are sensitive to convulsions like forotil. So we used, in one case, hyperthermia here, in the other case, on the left side, in the other case here, fluorotil, to induce seizures, comparing with the wild-type littermates. And so seizures can worsen the phenotype of these mice. And so you can transform with seizures a very mild GEF+ phenotype in a severe Dravet syndrome-like phenotype. But just if the mice have the mutation. If you induce seizures in wild-type mice, they don't get the severe phenotype. Okay? So you get the severe phenotype just if you have the mutation. And when you look at the mechanism, why do you have a severe phenotype, what we found here was that you get an increase of excitability of some excitatory neurons. So normally, as an initial functional effect, you don't see it. You don't see a modification of the property of excitatory neurons in these mice. But after the seizure protocol, you worsen the phenotype, you transform them in a Dravet syndrome animal model, basically, and you see hyperexcitability also in some glutamatergic excitatory neurons. So the conclusions are that both in vitro and in vivo models can provide information that can be valuable, but they should both be used with caution. And that experimental system can model different mechanisms, the initial functional effect, but also homeostatic responses and pathologic remodeling. And the SCN1A experimental models show different responses that can be induced by NaV1.1 dysfunction. The initial dysfunction of NaV1.1 can induce other dysfunctions that depends also on the phenotype of the animal. And so there is an interaction between the mutation, for example, and seizures in setting the severity of the phenotype. Thank you to all the people that collaborated to these studies, my lab in France, but also another lab in the same institute. Of course, the group of Silvana Franceschetti with Paolo Scalmani and Benedetta Terragni at the Besta Neurological Institute, and also Carolina Frassoni and Cristina Regondi. Chiara Verpelle and Carlo Sala for the iPS cells, Holger Lerse and Ulrike Hedrich for the study of the GEF+ animal model. Thank you.